Hello, my name is Georgios. I'm an undergraduate at Stanford studying chemical engineering, and I'm excited to share my research with you. Today, I will tell you about how we developed a model to isolate the effect of mechanical stress on epithelial fibroblast signaling. To begin, idiopathic pulmonary fibrosis, or IPF, is a progressive lung disease that affects over 3 million patients worldwide. Two key markers of the disease are increased lung scarring and excessive extracellular matrix deposition, thus making it difficult for patients to breathe. Looking at the cellular level, two main areas of investigation are one, epithelial fibroblast crosstalk that results in epithelial dysregulation and myofibroblast activation, and two, mechanical tissue stress as a result of stiff compliant tissue boundaries that again leads to myofibroblast activation. And despite the separate study of these two topics in the literature, the role of mechanical stress on modulating this crosstalk remains underexplored. And that is precisely the focus of my research. So to answer this question, we developed a custom 3D hydrogel model with two cellular layers. On the bottom, we have freshly FACS-sorted fibroblasts from mice challenged with bleomycin, a common IPF animal model. And on the top, we have in vitro cultured mouse epithelial cells. We allowed the cells to equilibrate inside the gel after encapsulation, applied a 10% biaxial strain, and then assessed the results using immunofluorescence and bulk RNA sequencing. First, looking at the whole gel in panel A, we see a clear spatial separation between the two cell populations. The fibroblasts were labeled with endogenous TD tomato, which you can see in red, and the epithelial cells with E-cadherin. We can see that there is a decrease in the E-cadherin expression as well between the control and the stretch group, which may be potentially indicative of reduced epithelial phenotype. Second, in panels B and C, we examined monoculture gels to assess the level of mechanoactivation. For the epithelial cells, we see some level of cellular alignment and increased alpha SMA expression. For the fibroblasts, we were especially interested in their mechanoactivation as their mechanoactivation leads to the excessive ECM deposition. So we optimized the equilibration time before stretching and the stretching time itself to achieve maximal mechanoactivation as quantified by the percentage of alpha SMA positive cells, as we can see in panel D. And the results were 48 hours of equilibration and 36 hours of stretching. Finally, we performed bulk RNA sequencing to assess differentially expressed genes in the control and stretch condition. And we found that several canonical mechanosensing markers, such as COL1A2, COL3A1, and ACTA2 were upregulated in the stretch condition, validating the model. And we also found that many lung disease markers that had been previously described were also upregulated in the stretch condition. Thus, we were able to conclude that our engineered 3D co-culture model demonstrates robust mechanical activation, providing us an in vitro platform that allows for the investigation of mechanobiological factors on epithelial fibroblast dysregulation. Looking forward, we're interested in continuing to examine the model for disease-relevant targets. With that, I want to thank you for your interest, and if you have any comments or questions, please reach out using my email at the bottom left of the poster. Thank you so much.